I'll just start with a very brief introduction. We are lucky enough to have two speakers today that they will be sharing the time slot. And they're both speaking about various types of neuroimaging biomarkers as they're used in Alzheimer's disease research. And both researchers are in the Department of Neurosciences and are collaborating with the ADRC and recruiting from our pool of participants from our longitudinal cohort. So this is an opportunity for all of our colleagues to hear more about both of their projects. So first off, we have Dr. Emilie Reas, and she is in the Department of Neurosciences, and she is researching brain microstructural biomarkers of Alzheimer's disease and cognitive aging. She's going to tell us more about that today. And then she will be followed by Dr. Sarah Banks, who's also in the Department of Neurosciences as a neuropsychologist, and she will be presenting about what can PET techniques tell us about Alzheimer's disease, looking at tau pathology. So both of those researchers, we are very pleased to have you and very grateful for your participation in our Lunch and Learn. And with that said, we'll make sure that we're leaving some time at the end for questions. So if you could hold those until the end, that would be great, or add them to the chat, and then we can review those systematically at the end. And I will mute myself and give you the floor. Thank you so much.
Okay, thanks, Christina. And thanks for the opportunity to speak today. So let me see if I can share my screen here. So today I would like to walk you through some of our prior research using the diffusion MRI technique restriction spectrum imaging to examine brain microstructural changes in Alzheimer's disease and cognitive aging. Okay, so I'd like to start with just a little bit of background. Now, some of you may be familiar with this model of Alzheimer's disease, presenting the time course of various Alzheimer's disease biomarkers. But this present model posits that particular brain changes occur along a characteristic time course of disease progression. Initially, the toxic proteins amyloid and tau accumulate in the brain, and this may occur decades before symptoms appear. After that, structural changes appear. These include volume loss, cortical thinning, and degeneration of the white matter pathways in the brain. And these structural changes can be visualized using magnetic resonance imaging, or MRI. After these structural changes, cognitive impairment begins to emerge, with the most notable deficits in memory, followed later by impairment in other domains such as executive function and verbal fluency. And ultimately, functional and behavioral disturbances appear that characterize full-blown dementia. Now, of these biomarkers, MRI measures of brain atrophy have become one of the most valuable and widely used markers of disease progression and disease risk. MRI is particularly valuable for several reasons. First off, it's non-invasive. So as I showed here, the earlier changes tend to be accumulation of amyloid or tau. However, measurement of these pathological proteins typically requires a painful lumbar puncture or PET scans that Sarah will talk about later, which carries a risk of low exposure to radiation. So MRI is non-invasive. It's easy to perform in the clinic. It's been shown to be highly sensitive to clinical outcomes, cognitive decline, and disease progression. It correlates very well with cognitive measures. And the patterns of cortical thinning and volume loss tend to be unique and specific to Alzheimer's disease. So we're able to be more confident in one's diagnosis of Alzheimer's disease versus other forms of dementia or neurological disease. However, MRI also has some limitations. Particularly, it is blind to microstructural changes that occur early in Alzheimer's disease and likely precede these gross measures of atrophy. For example, smaller scale changes at the cellular level in the brain in the early stages of Alzheimer's disease include things like synapse loss, reduced axonal density, loss of dendritic branching, demyelination, cell shrinkage, and standard structural MRI can't detect those changes. So that's where diffusion MRI comes in. So some of you may be familiar with the technique diffusion tensor imaging, which is the most common form of diffusion MRI. But basically, diffusion MRI measures the magnitude and the direction of water diffusion that allows us to estimate different parameters of tissue microstructure. This is possible because diffusion patterns differ according to cell morphology. So for example, in free water spaces such as the cerebrospinal fluid or CSF, water will diffuse very quickly and in all different directions or in an isotropic manner. Conversely, in small constrained spaces like in cell bodies, diffusion will be very restricted and will occur on slow timescales. Furthermore, within long cylindrical spaces like axons and dendrites, that restricted diffusion will occur primarily along the axis of the axon or dendrite. So basically, we are allowed to, we are enabled to infer tissue microstructure from these diffusion patterns. Now, there are various forms of diffusion MRI out there. As I mentioned, DTI is the most common. Here at UCSD, we have been using restriction spectrum imaging, which was developed by Anders Dale and colleagues. I won't go into all the nitty-gritty details because that will take an entire talk. But basically, it's a multi-direction high B-value acquisition combined with a model of diffusion orientation across a spectrum of length scales. So this model allows us to separate out different compartments of water diffusion: restricted diffusion at the smallest scale, hindered at the intermediate scale, and free water at the largest scale. The model also allows us to resolve crossing fibers or areas where axons intersect, which is a very valuable measure. So from this model, we are able to compute a whole range of different microstructural parameters that we have found useful in detecting signs of cognitive impairment or brain aging. And so I'll just briefly walk you through 5 of the measures that we found most useful. So starting at the smallest scale of restricted diffusion, again, this most likely corresponds to diffusion within cell bodies. And we have 3 different measures for restricted diffusion: restricted isotropic, which would correspond to small cell bodies; restricted oriented, reflecting axons and dendrites; and crossing fibers, where those axons or dendrites cross. At the intermediate scale, we have a measure of hindered isotropic diffusion, which we believe would correspond to larger cell bodies or the extracellular space. And then at the very largest scale here, we have isotropic free water, which would correspond to free water spaces such as the cerebrospinal fluid. Okay, so over the past several years, we have used this RSI technique to examine whether we can to identify microstructural changes in both early Alzheimer's disease and in normal cognitive aging. And we set out to address 4 main questions. The first question is, are these RSI measures sensitive to cognitive impairment in the early stages of Alzheimer's disease, or MCI? So to address this question, we recruited 56 participants from the ADRC longitudinal study, age 63 to 93, including 31 healthy controls and 25 individuals with either amnestic MCI or mild AD. And because the mild AD participants were so mild and did not differ on cognitive or imaging measures from our MCI group, we combined the MCI and AD participants together for analysis. This group of participants attended a visit in which they completed RSI and structural MRI. They also underwent comprehensive neuropsychological testing through the ADRC, and a subset of 38 participants underwent lumbar puncture for measurement of CSF amyloid beta. Now, as you can see here, participants did not differ in terms of age, education, or IQ. However, the impaired participants had a greater proportion of men, and they performed worse than controls on a range of tests measuring dementia severity, functional ability, logical memory, and episodic verbal memory. This is to be expected. Oops. Oops. Okay. So first, we just wanted to determine whether we could see any differences in our RSI measures based on diagnosis. And indeed, as you can see here, each of these rows is showing a different RSI measure. The top 3 rows are showing restricted measures. The bottom row is showing isotropic free diffusion. And as you can see, in a number of white matter fiber tracts, as well as in the hippocampus and the entorhinal cortex, impaired participants had lower restricted diffusion and higher isotropic free water than healthy controls. In addition, we wanted to see if there was a relationship with cognitive performance and brain microstructure. And I know this is a lot of data here, but this is simply showing Pearson's correlations between restricted diffusion in this top panel, isotropic diffusion in this bottom panel, and performance on various tests, the dementia rating scale, which is just a measure of dementia severity, and then 3 measures of delayed memory. And as you can see from all these starred values, those are significant values, there were very strong correlations between higher restricted diffusion and better cognitive performance, lower isotropic free diffusion and better cognitive performance on a range of white matter fibers, as well as in the hippocampus and the entorhinal cortex. So this, preliminary data was very encouraging, showing that we were seeing very robust associations between brain microstructure and cognitive impairment, even at the mild stages of Alzheimer's disease. So as I mentioned in my first slide, the earliest biomarker change is an elevation in amyloid pathology, accumulation of amyloid beta. So since amyloid is really the classic hallmark of Alzheimer's disease pathology, we wanted to assess whether microstructural changes in mild cognitive impairment also correlated with Alzheimer's pathology. And we found very robust, consistent correlations between increased amyloid levels, or actually lower CSF amyloid, which corresponds to greater brain burden, and altered brain microstructure. So these plots here are showing, on the left here, restricted diffusion across all white matter fibers in the brain. In the middle, free water across all cortical gray matter. And then on the right, free water across all cortical white matter. And as you can see, there, even in this relatively small sample of only 38 participants, we see relatively strong correlations between greater amyloid burden and microstructural injury. Okay, so so far we've seen that these RSI measures of reduced intracellular diffusion and increased free water expansion of the CSF space correspond with cognitive impairment and AD pathology. However, a more valuable biomarker than just being able to detect cross-sectional differences is if we can predict whether a participant is going to progress along the disease course or demonstrate cognitive decline, because really interventions will be most valuable before the disease becomes more severe and cognitive decline progresses. So to address this question, we brought participants back in approximately 2 years later. 47 participants returned for follow-up cognitive testing and 38 returned for follow-up imaging. And we were interested in seeing if our RSI measures at baseline could predict subsequent cognitive decline, or if the rate of change in our RSI measures would correlate with rates of cognitive decline. And yes, indeed, here on the left panel, we see that hippocampal restricted isotropic diffusion at baseline, or I should say lower restricted isotropic diffusion at baseline predicted more rapid decline in a measure of verbal fluency. In addition, the magnitude of change in restricted isotropic diffusion in both the hippocampus and the entorhinal cortex also correlated with more rapid decline in dementia severity and in executive function. So this was actually really exciting to find that these RSI measures aren't just sensitive to existing cognitive deficits, but they may be valuable biomarkers for predicting disease progression. Okay, so lastly, we know that the strongest risk factor for Alzheimer's disease is age. In addition, we're developing a greater understanding of some of the other risk factors for Alzheimer's disease, which include various lifestyle, health factors, but there's emerging evidence that sex is one of the stronger risk factors for Alzheimer's disease as well. So in some of our more recent studies, we wanted to understand the role of both age and sex in microstructural changes that may predispose somebody to more rapid cognitive decline in even normal aging, perhaps in the preclinical stages of Alzheimer's disease. So to address this question, well, I should say Linda McAvoy, she has led this study of the Rancho Bernardo study, she has led the imaging component of the Rancho Bernardo study. For those of you who are unfamiliar with this study, it's a longitudinal study that began here at UCSD in the 1970s. And for the last visit, Linda McAvoy brought participants in for a final assessment where they underwent RSI and MRI scanning. This study included 90 women and 57 men, aged 56 to 99, with an average age of 77. And we found very robust correlations between older age and signs of microstructural damage in this cohort. And let me remind you, these are all community dwelling, relatively healthy older adults. None of them were diagnosed with dementia. So this is a relatively healthy sample. And these plots here are just showing correlations between age and white matter microstructure across all fiber tracts. And we're seeing that older age strongly correlates with reduced restricted diffusion and increased isotropic free water. So as I mentioned before, sex is another strong risk factor for Alzheimer's disease, with women having a significantly greater chance of developing Alzheimer's disease in older age. So we next compared these correlations between age and microstructure between men and women to see if we saw any sex differences in this age-dependent risk for microstructural damage. And I hope this plot shows you how striking the difference is, but we have women here on the left, men on the right. Each of these rows is showing a different RSI measure, and the colors are just showing areas of significant correlations between age and the RSI metric. The blue colors are showing negative correlations, the warm colors are showing positive correlations, and as you can see, our women are showing much stronger correlations between older age and lower restricted diffusion, higher isotropic free water than men. Now you may note that we had a much larger sample of women than men. So to examine whether this sex difference could just be due to differences in statistical power, we created a match sample of 57 women of the same age as men, and basically our results did not change. So there's this striking difference where women are showing a much stronger detrimental effect of aging on brain microstructure, which is very interesting in light of the greater risk of women for developing dementia. Okay, so our next question was, all right, so we see these signs of microstructural injury, even in normal aging. Does that translate to cognitive impairment? These participants of the Rancho Bernardo study had also undergone a range of cognitive tests. And so we performed correlations between our RSI measures and performance on these cognitive tests. Here, we're showing correlations for women on the top panel, men on the bottom panel. Each row is showing a different cognitive measure, and I won't walk you through all the details for the sake of time, but as you can see, there are very robust correlations between our restricted, hindered, and free water measures, and a range of cognitive tests, global cognitive function, executive function, and memory, slightly stronger correlations for women than for men. So lastly, we knew so far that we are seeing strong correlations between age and microstructure. We're also seeing correlations between microstructure and cognitive function. So the obvious question is, well, are these microstructural changes actually mediating effects of aging on cognitive decline? So to address that question, we performed a formal mediation analysis. And yes, indeed, we found that various microstructural parameters mediated the negative effects of age on cognitive impairment. Interestingly, we found that fiber microstructure mediated effects of age on visual spatial memory for both women and men. However, we observed some interesting sex differences as well, where hippocampal microstructure mediated effects of age on verbal episodic memory, and white matter microstructure mediated effects of age on executive function for men. So again, this is kind of reinforcing the suggestion that men and women undergo different patterns of cytoarchitectural brain aging, and that may contribute to the different patterns of cognitive decline in older age. So to summarize, we found that a range of RSI metrics that we believe reflect reduced neurite density, cell loss, or cell shrink. Were able to discriminate between cognitively impaired and healthy older adults. They also correlated with amyloid burden. They predicted rates of cognitive decline and disease progression. And they also mediated effects of normal aging on cognitive function with distinct age-related changes for women and for men. So to conclude, we believe that RSI may be useful for detecting subtle cytoarchitectural brain changes that occur early in Alzheimer's disease, or even in just normal age-related cognitive decline, and that RSI may be a valuable biomarker for monitoring therapeutic outcomes in aging or Alzheimer's disease. So with that, I will pass it over to Sarah Banks, who I think will be talking a little bit more about the imaging technique PET and how we can use that to assess amyloid and tau.
Thanks, Emily. Nice talk. So thank you very much, Christina, for inviting me to talk, and everyone for listening. I'm going to talk a little bit about PET and what it can tell us about Alzheimer's disease. So I thought we would just go over the basics of what is PET and why it's important in studying AD, and then go into some of the sort of newer and what I think are exciting elements. And this is a PET scanner. So as I think most people on the call probably know only too well, AD begins as a disease in the cells. So before there are structural changes, and by structural changes I'm really talking about sort of hippocampal atrophy and the grosser atrophy rather than the changes that Emilie was talking about, there are changes at the cellular level which can't really be picked up by MRI. So PET works when a substance is injected into the body which binds to some cells and not others, or some proteins and not others. Of radioactive labeling, And the PET camera then picks up on that label. So it shows where the cells of interest are located in the living person. And we can learn how much of that cell you have, but also where it is in the brain. And those two things are very important in Alzheimer's disease. And of course, PET is very useful in AD, but it's also used in other diseases where cellular changes are very important, like notably cancer. So in Alzheimer's disease, there are two main proteins or culprits that lead, we think, to the disease and the dementia. One is amyloid, and that starts very early on, probably 10 to 15 years before any symptoms are shown. We see this steady buildup of amyloid, which is in these kind of beigey colors at the top, and it becomes quite diffuse quite quickly throughout the brain. So probably not until somewhere in this area are we seeing any symptoms. Tau, I think, is in many ways more interesting because although it starts relatively early in these very subscribed areas of the brain, in people, especially people who are amyloid positive, who have a lot of amyloid buildup, there is this spread, and the spread kind of goes along with the cognitive deficits. So the spread starts in the entorhinal and hippocampal regions, which are really important in memory. And then it kind of follows this arrow forward into the frontal lobes, and then people get kind of this executive syndrome where they can't plan as well, they can't switch tasks as well, and then it continues into the more posterior parts of the brain, and then you get visuospatial problems. And at this stage, when the tau is quite widespread in the brain, that's normally when you see the kind of full-fledged dementia and people aren't able to take care of themselves so well. So up until a few years ago, we couldn't really learn very much about amyloid and tau until we waited for post-mortem studies. And obviously at that point, it's not ideal. The person has to be dead. So the PET revolution in Alzheimer's disease has really been pretty amazing, and learning more about what's going on early in the process, not only how much they have, which we could get from CSF before, but also where it is in the brain, which for tau in particular is really important. So this is kind of just the stages that we see. So the amyloid beta protein builds up at least 10 years before the symptoms and can be seen with PET. And then the pathological tau seems to build up about a year before subtle symptoms. So a lot of us who are over 40 will have some pathological tau in our brains. Like if I got hit by a bus tomorrow, you'd probably find a little bit of tau in my entorhinal region. But for it to move forward, that's normally when the pathology starts to happen. And that generally happens when you're a bit older. It starts in the memory centers and then spreads as we went over. And again, that can be seen on PET. The cell death and brain atrophy, so when we're getting down to hippocampal atrophy and atrophy that you can see on just a regular MRI like we use in clinic, that seems to happen as a result of tau. So it's kind of downstream of the tau process. It happens after the symptoms begin, and it can be seen on MRI, but it's kind of later in the process. So I'm going to start by talking for a minute or two about amyloid PET. So this has been around experimentally since about 2004. These days, you can get one as a clinical scan, but the guidelines are to not use it on somebody who doesn't have symptoms, and this will be in clinic. So if Dr. Galasco saw a patient who really wanted tau PET and that patient had memory symptoms, he could order it. If that patient didn't have memory or cognitive symptoms, the guidelines, the sort of ethical guidelines, would be just to not do it at that point. It binds, the PET ligand binds to this early form of amyloid, which happens actually before the plaques that we hear about a lot in Alzheimer's disease forms, and it's probably the most toxic component of the amyloid kind of cascade. Flabetapir scans, so flabetapir is the name of the ligand, the medication that gets injected into somebody. It's one of a few different ones, which shows up with amyloid in the brain. So reading flabetapir scans is actually kind of simplistic. So this person had no cognitive symptoms and has a negative scan, has this big white ring and the kind of fuzzy area, and then the darker area is inside. This person with a positive scan, you can see the dark area goes right up to the edge here of the brain. This person had mild cognitive impairment. They also had reduced hippocampal volume, so they were a little bit further along. So you see this decreased gray-white contrast in the positive scans, where more radio uptake, so more uptake of this radioactive medication is happening closer to the cortex or the edge of the brain. So at the kind of gold standard is these qualitative reads, where somebody who's qualified to read them, and the qualification isn't a very complicated thing to get, can say whether or not someone is negative or positive. And it actually agrees pretty well with more qualitative assessments, quantitative assessments, sorry, that you can do by doing the sort of measuring the darkness. And these pictures were taken from one of Rahul Desikan's papers. So some pros and cons of amyloid PET. One of the big pros these days is it's been approved by the FDA, so you can actually get it done without having too many complications. But at the moment, it's not covered by Medicare or any insurance carriers. And it's not cheap. It's about $7,000 a scan. So you have to really want it and be able to afford it. The amyloid is present very early in the process, before the clinical symptoms occur, but it's also very diffuse. So it doesn't locate to the areas that are important in the cognitive processes that we might think are important, especially in the research area. And another really important thing is, if you're only going to get a positive scan 10 to 15 years before your symptoms, and you're 75 when you get your scan, well, you have to live till you're 85 or 90 to get those symptoms. So, of course, a lot of people will die within that age range, and there's a lot of ethical conversation about whether it's right to do these kind of scans on people who don't have clinical symptoms, because what happens to that person when they know that Alzheimer's is in their future, and how do they change their life, and so on. So just something that people are thinking about that's really important. Another pro, of course, is it's quite easy to do those scans. So tau PET, tau pathology in Alzheimer's disease is primarily tangles, fibrillary tangles, which accumulate in the cell bodies. And tau is an essential protein early on, before it becomes pathological, for stabilizing microtubules, an important part of the cell. But this is disrupted, which causes misfolding, and then there's that kind of tangle in the brain. And tau accumulation begins in the locus coeruleus, right in the center of the brain, and then it continues into the medial temporal lobes, where it begins to correlate with those underlying cognitive changes. So to talk a little bit about tau and tau pathology. So here at sort of stage A, this in the background, a nice healthy brain cells, and it's stabilizing the microtubules of the cell. So it's actually a really important protein. And then the oligomers of tau tend to build up in the cells, and we think that they propagate cell to cell. So something has to make tau spread, and they think that it's this cell-to-cell propagation, and it's areas which are well connected, probably because they're important cognitively or functionally together, that causes these relationships, which create somehow a space for them to share tau, pathological tau. So it then begins to kind of build up in the cell body, these oligomers, and that's where we get the tangles, which prevents the normal functioning of the cell. So the cell is all clotted up with this neurofibrillary tau. It doesn't work anymore. And then later on, the neurite itself dies, leaving only this kind of what's called a ghost tangle. So it's kind of the residual of the tangle. And this is where we see tau PET, the tau PET ligands kind of attaching. So one of the reasons that tau PET is interesting and important in Alzheimer's disease is Alzheimer's doesn't always follow the same cognitive profile. Most people, the great majority who get Alzheimer's disease, will have amnestic Alzheimer's disease. So this is where we get memory loss early on, and then it spreads around. So this is somebody who has early amnestic Alzheimer's disease. And you can see there's a lot of tau in the medial temporal lobe, a little bit in the parietal lobe, and not a lot somewhere else. Some people, and this is rare, but it certainly happens, have something called the logopenic variant of primary progressive aphasia, which is an unusual form of Alzheimer's disease, which initially just presents with a lack of words. So people start to really stumble on their words. If you test them appropriately, you can find that they have intact memory, especially for visuospatial things, but words are really problematic for them. And those people, if you look at the left hemisphere compared with the right hemisphere, have a big buildup in the left hemisphere language areas and much less on the right. Another unusual kind of Alzheimer's disease is posterior cortical atrophy, and this is where somebody has visuospatial problems early on as opposed to memory. And you can see in that case you get tau buildup in the posterior regions in the early stages. And then there's other unusual forms as well, and this group just did a really nice job of showing how tau PET really reflects where we would expect there to be underlying tau in the brain, but rather than waiting till autopsy, we can now see that during life. I'm very interested as a neuropsychologist in cognition, so memory and other thinking abilities, and to me it's really great to have this tool to use to look at the changes that are directly related to cognition. And this was a study that we did a couple of years ago now. Dino Digma was the first author, and we were looking at how tau PET in the ADNI study, of which I believe we're a site, looks at how the tau PET connects with different cognitive tests. So unfortunately, in ADNI, in most studies, they only test verbal memory. There's no visuospatial memory. But what you can see is that if you look at this verbal learning test in relation to tau PET, we see that it's an extensive relationship in the temporal and parietal regions as well as the frontal regions, more so in the left. And again, it's a verbal task, so you expect the left hemisphere to be more involved in that. And then here, this is the MINT, a language task that everyone here is quite familiar with. Again, it's a very left hemisphere correlation, which you'd expect, whereas something that's more processing speed, more diffuse, goes bilaterally kind of everywhere. So we're building off of that research. This actually shows the relationship between tau PET and MRI. So with the kind of MRI we get in ADNI, we can look at cortical thickness, so how atrophied or not atrophied the cortex is. And what we did here is compare, this is what you saw in the previous scan, the tau PET in relation to these different cognitive scores. And then here on the right is the cortical thickness in comparison with those different scores. And you can see how much more kind of sensitive the tau PET is to the performance on, say, the adult learning compared with cortical thickness. So tau PET is a really exciting tool for those of us who do neuroimaging. There's also a relationship, as you would expect. Emilie showed you those curves earlier, and we were talking earlier about how amyloid and tau happen kind of earlier and we get atrophy later. So this group up in Berkeley looked at tau PET and how that relates to longitudinal atrophy. And you'll see that regions of the brain where there was extensive tau PET early on show more atrophy in the future. So it kind of predicts atrophy in the future. So how does this all, how does this all connect? So early on you get this kind of diffuse cortical amyloid beta and a little bit of tau PET in this restricted region. But when the two of them combine, so when someone's amyloid positive, there begins to be this spread of tau in these different regions. And ultimately that ends up causing neurodegeneration or atrophy and dementia. But there are lots of things that can have an impact on that. So there's things like sleep and increased neural activity and physical activity and genetics that have an impact on either amyloid or tau or some other process which is in between cerebrovascular factors. So in our group in the Human Memory Lab, we're interested in looking at some of these factors and how they might explain some of these relationships. Like Emilie, I'm very interested in sex differences. We've known for a long time that women demonstrate a lifelong advantage on verbal tasks. So learning lists of words, etc., etc. I'll give you an example in a minute. And it turns out that women have a reserve against verbal memory loss. So we can get a lot further into the disease before we start declining. And once we start declining, though, we tend to decline quicker. So it's not kind of a linear relationship, it's a complicated relationship. What we found, and this is consistent with both autopsy studies and CSF studies, is that women have more tau in their brain at a comparable level of the disease. So in mild cognitive impairment, where men and women are relatively equal in terms of functional impairment and other cognitive problems, women have just a lot more tau. So this part of the brain is a lot more yellow, showing a higher level of buildup of tau compared with men at the same stage. The distribution is generally very similar. It's just that there's more of it in the women. How do we test verbal memory? I think a lot of people in this call are very familiar with these things, but the two main ways that we test verbal memory are list learning tasks. So you might read somebody a group of nouns and then ask them to repeat the ones they remembered, and then do that three, four, or five times, depending on which one you're using. And then a little while later, you say, remember that list, and the person moans and then tells you the few words that they maybe remembered. And then the other way is stories. So we might read somebody a little story or a more long story if we're being really mean, and then we'll ask them to tell us which elements of the story they remembered, and then again 20-30 minutes later you kind of spring it on them. So we find that these kind of learning and memory tests, verbally based, are quite different in how they are related to tau in men and women. So the Reas-free verbal learning test, this is the particular learning test that's used in ADNI, is, if you look at the delayed recall, there's a lot more extensive relationship in women of tau PET to delayed recall. And similarly, in logical memory, which is a story memory test, we see a stronger relationship in women than men. And actually, when we compare those directly, you can see the red areas are the areas that are statistically stronger in women, and the yellow areas, of which there are not a lot, and they tend to be in different areas, sort of frontal and posterior there, where we see stronger relationships in men than women. So women can, this is really showing that women can have a lot more tau in their brain and yet perform at a very similar level. The men and the women in this study did not differ significantly on how well they did on the test, but despite this, women seem to have a lot more tau in those regions. The next step for myself and Erin Sunderman, who some of you know, is the Women Inflammation and Tau study. So we're going back to some of these factors and looking at how women specifically, we're trying to get some funding to look at men too, but we're going to start with women, how these modifiable or potentially modifiable risk factors might contribute to tau burden and accumulation. So we know that in women, but not men, inflammation, neuroinflammation, so inflammatory processes in the brain seem to spur some of that tau buildup. And we're interested in seeing how activities that impact inflammation, so sleep, physical activity, and diet, are related to neuroinflammation, and how those are related to tau. And ultimately, of course, we'd like to know if we can interrupt that process, and that interruption or how we affect that might be different in men and women. So this is our fancy new logo for the WITS study. And that's it from me. Thank you. Okay, I think we have some time for questions if anybody has any.
Question in the chat. Dr. Brewer took the liberty of answering one, but it looked like a few followed. As far as questions, it looks like on the RSI, there was a question about whether that imaging technique, oops, it's hopping around on me, sorry, is easily implemented across multiple clinical sites, or whether it requires an advanced magnet and imaging procedures and analysis. And ultimately, the question is just wondering how broadly it could be applied, or whether sites need to be relatively sophisticated.
Yeah, so that's a great question in terms of kind of the practical implementation of this. So the RSI protocol can be implemented on any magnet. So we have it here on multiple 3T scanners at UCSD at different centers. I know we have a collaboration at UC Davis where they also have the RSI protocol there. Really, all you need to do, you need to know the particular MRI parameters to set up your protocol, but it doesn't require a fancy machine or anything. The complication, or I guess the hardest part, would just be getting your image processing set up. So we have a system set up here with Anders Dale and Don Hagler. They set it up for us, and we have this stream where we can process the data. So realistically, we could probably set up that same processing stream at a different site, or the data could get sent to us and we could process it. So I don't think there would be any real limitations to having this technique set up at different sites. I should mention that RSI is not the only advanced diffusion MRI technique out there. Another popular one is NADI. I won't go into the details of that, but different sites have their preferred diffusion imaging method. There are advantages and disadvantages to using one over the other, but we like RSI.
If I don't cover all aspects of your question based on the chat, please feel free to hop in and ask. I'm just trying to keep it moving. The next question was, are there reasons why a patient wouldn't be able to do PET?
Well, I think, if they're not comfortable with having the injection, or if they're claustrophobic, those would certainly be contraindications to PET. And then it's why they're having it done, and whether or not it's appropriate.
Okay. Do we know which inflammatory pathways are lit up in women?
Well, we're getting some idea. So with one of the things that we're finding, which I think is really interesting, is that there's not necessarily more inflammation in women than men, and this is certain cytokines such as IL-6, TNF-alpha, but the relationships between inflammation and tau are much stronger in women than they are with men. So a guy can have a bunch of IL-6 in his CSF and not have that much tau, but if a woman has a lot of IL-6, then she has a lot of tau. So we're not quite sure why that is, but there's definitely sort of different relationships, and we're looking at that in some papers that we're writing up right now, again, based on ADNI.
Great. It looks like the last one that was submitted through the chat is, do you think sex hormone contribute to the AD in the women? I mean, since over 65 women will be menopause.
So that's an excellent question, and we just published a paper on this. So yes, estrogens and estradiol kind of hit not rock bottom, but they get very, very low and hard to measure in postmenopausal women. What doesn't hit rock bottom necessarily in women, which shows a lot more variation, is testosterone. And a lot of the studies that have looked at testosterone have totally neglected women. They're like, that's a male hormone, let's just ignore the women. Well, actually what we're finding is that testosterone is related to tau in women, and it's also related to inflammation. So we're looking very closely at how tau might relate to physical activity, and how it might help explain that. So people, just to backtrack a second, people who have low testosterone, and those tend to be women, but it can also be men with low testosterone, tend to have more tau and also more inflammation.
Great. There aren't any other questions through the chat, but if anybody else thought of a question while you were hearing responses, please feel free to unmute yourself. Or
I have a quick question for Emilie. Emilie, so when you're looking at the microstructural properties, right, that in different regions that are predictive of like longitudinally on some measures, how much does the diffusion parameter contribute in comparison to like just like volume of that same region?
That's a really important question, actually, and that's something that we've looked at in our prior studies. And we had assumed that a large proportion of the variability in diffusion would be attributable to volume loss, particularly for measures like the free water, isotropic free water, which would be driven by atrophy. To our surprise, when we redid these analyses controlling for like cortical thickness or hippocampal volume, to account for that, we saw still very robust correlations between our RSI measures and cognitive variables or age or whatever you're looking at. So that to us suggests that the diffusion patterns that we're picking up on are strongly driven by cellular changes, not just shrinkage, volume loss. But that's a really important thing to control for, because I mean, what's the value if we're just kind of replicating what you can do in a standard structural MRI? Yeah, so I do think that we're picking up on actual cell microstructure and not just brain shrinkage.
Awesome, thanks.
another question got submitted about whether there were limitations for women that had a history of breast cancer treatment and doing PET scans.
I think we look at how much radiation a woman has, or anyone coming up for a PET scan has had in that year, if that was the concern with radiation therapy. Certainly in our studies, we don't exclude people who previously had breast cancer, but we would want to know about it for sure. So.
But thank you for joining us, and thanks so much for taking the time to prepare two excellent presentations. We're very grateful.
Thanks, everyone.
All right. Bye. Bye. Thank you.